Last but not least, someone has to go last, and there's Peter Janssen, who is a principal scientist at Ag Research. He's made a career out of trying to culture the unculturable microbes. Been in New Zealand since 2007 and is an invaluable scientist across a whole range of programs, but he jointly leads the methanogen vaccine program with Neil Wedlock, and we're really looking forward to this, and we're hoping on the way out you've got the vaccines ready for us, that you've made the discovery and we can all have it on the way out, Peter.
Do you have a methane problem, Harry? Yeah.
I wouldn't answer on the grounds that it may incriminate me.
Yeah, so I'm Peter, and just to clarify, I was born in New Zealand, and I came back to New Zealand in about 2007 when Harry was my boss then, and he's still my boss now in a different way. So I'm part of Neil Wedlock's team, and we're developing a vaccine to reduce methane emissions from ruminant animals. This is not a widget game. This is not about going to the shelf or to the literature and finding something and adapting it. This is about doing the science. It's a very science-intensive program because there is no such thing on the market. There's no such thing being done before, so we're trying to do something quite new. So what makes it attractive against that science challenge is that we're talking about a low-frequency intervention, maybe vaccinate once a year. It makes it applicable to New Zealand's pastoral farming systems. Vaccination is largely acceptable, probably to the consumer as well as to farmers. Virtually no risk of residues in saleable products, and it's also auditable. If you vaccinate 1,000 cows and you know there's a 30% reduction, then you can calculate how much methane you've reduced. We've talked about this bit before, so I won't go into all the details, but a vaccine produced in New Zealand would be globally applicable because the methane-producing microbes are the same right across the globe, every rumen you look at. So we've done some experiments where we've seen small and not statistically significant reductions of methane using experimental vaccines. We're repeating that just to make sure that's not just a statistical fluke, which they always can be. But we're aiming for something like a 30%, maybe more, so we can produce something that's meaningful and can be used on farms that are meaningful, to make a meaningful reduction. So a vaccine consists of a number of components. There's the antigens. That's the part that you want the immune system to react to. You want a part of the organism that you're trying to control. You add an adjuvant that causes the immune system to respond to that and produce antibodies and an immune response. And then there's a way of vaccinating, how you administer the vaccine, timing, and so on. In our vaccine, we're relying on antibodies entering the rumen via the saliva. So ruminant animals produce copious amounts of saliva. Cattle produce 100 to 300 litres a day. We know there are enough antibodies in that saliva to have an effect that is not just total antibodies, but antibodies that we induce by vaccination. And so those antibodies enter the rumen. They need there to bind onto that antigen that you've been targeting, and in such a way that they interfere with the activity of the methanogens, reducing their ability to grow, survive in the rumen, and produce methane. So we know that these antibodies interact with methanogens in test tubes. We've shown that a long time ago. We're really interested in whether they interact with methanogens in the rumen. And so this slide here, this is a very early preliminary or early results. If you look at the yellow line, the orange line, that's just showing as you go along, we can get something like 40% of methanogens in a rumen sample to bind antibodies to an experimental vaccine. So that's just first off a very early stage experimental vaccine. 40% of the methanogens will bind to, or antibodies will bind to those methanogens in the native populations in the rumen. There's also, if you notice the blue line, these are unvaccinated control animals they also contain antibodies already to methanogens, which is hardly surprising because the methanogens are part of the system, so it's not surprising they've made an immune response. But it also tells us what we're trying to do is we're trying to enhance and steer something that's already occurring naturally. We're not trying to make something happen that's not natural. We've also shown in another study, I'm not going to go into the detail, that if you vaccinate with individual cells from individual strains of methanogens, that the antibodies produced will cross-react with a wide range of methanogens in the rumen. That's the native populations. So there's real promise here for a vaccine that can control a large proportion of the methanogens in the rumen. And as I said, these are all early stage vaccines. So as well as figuring out how to get the vaccine to work and bind to rumen methanogens, we're interested in refining the vaccine itself. And I'm just going to give you a very small example of the work we're doing. If you can imagine a protein, so this is a protein maybe that we want to target, and it consists as a string of amino acids, like a string of beads, and it's all folded up into a very specific shape. If we now imagine unraveling that, so that's the line down here, and we're looking at these Ps, the things labelled Ps, these rows are different just regions along the length of that amino acid chain. And the red colour indicates the strength of the immune response we get to that part of the protein in 12 different animals. So we see two things here. We see that some regions of these antigens are much more immunogenic, so they induce a much stronger immune response. And some parts are weak and are just sort of wasting your time trying to vaccinate against those parts. So that's important. And the second part is there's a lot of animal-to-animal variation, which is a challenge that we need to overcome because that in the background is going to reduce the response that you see in a group of animals. We've done that for, we've got a set of 9 proteins, 9 antigens that we're looking at in detail at the moment, and we've done that for multiple, and here are some other examples of studies on those, and you can see the same pattern, strong immunogenic regions and also animal-to-animal variation. Another aspect of it is when those antibodies bind onto the antigen in the rumen, do they just bind and then fall off and then bind and then fall off and actually not doing being active for most of the time? Can we increase the strength of that interaction, which we call avidity? And here's, again, which is an early experimental vaccine, but you can see the grey bars and the dots of the data points. They represent the avidity when the vaccine is applied in a certain way, and you can see that over time the avidity actually increases. But quite unexpectedly, you'll also see that the variation, that the individual animals are the black points, you can see that the variation of the avidity decreases. So we can see that you can, at least in this one aspect, you can actually overcome that animal-to-animal variation and perhaps bring them all up to the high level that you want. So in addition to this, in addition to, these are just some little glimpses into the data. We're also trying to continue our studies to find the best, most effective antigens to have in a vaccine. One of the things we're trying to do as well is how do you get those that you have to produce in the laboratory, how do you get those antigens that are synthetically made to mimic the native structure so that the antibodies recognise the native? It's no good if they recognise something synthetic that doesn't look like the native, they're not going to bind very well. So that's an important part of our program. The vaccination strategy and timing can be very important, so we've got experiments going on that. We have looked at adjuvants and we'd really like to go back to looking at adjuvants for much stronger immune responses. And I should say in that area, in that broader area, we're very happy to have heard the announcement from the Minister that there's some additional funding coming into an allied area along this approach. And also we're very interested in seeing how we can direct more of the immune response into the saliva. It's all very good, the animal are mounting an immune response, but we want that in the rumen. It has to enter via the saliva, so that's a very important additional point. And so what we do in our research is we stack these gains, and we do each experiment, each series of trials stacks the gains made in the previous ones. And so it's by stacking all these gains that we will reach that 30% target that we've set ourselves for a vaccine that we feel can be useful for farmers. So the research The research is being done at AgResearch in Palmerston North on the whole. We also have collaborators at the University of Victoria, University of Wellington, and the Moredun Research Institute, the Sydney University of Technology. We're very lucky to have the expertise of Jonah Roberts for flow cytometry work. And very important element, and I saw Emily Parker here before, who's on our methane vaccine think tank. This is our science advisory group who ask us hard questions, review our program, and generally are very useful for advice, either as a group or we go to individuals or subsets of that group to get advice on how to design experiments or whether things, research ideas, bounce research ideas off them, I suppose. And over the years, the research has been funded by the PGGRC, who incidentally also manage the intellectual property that we've developed, in case anyone's interested in talking to them. Individually as well by organisations that were also in the PGGRC, Beef and Lamb New Zealand, Dairy NZ, Deer Industry New Zealand, Fonterra and AgResearch, the New Zealand Agricultural Greenhouse Gas Research Centre, and of course we've also been supported by the GRA, particularly with some of the method development, and you might have noticed occasionally there was a comment like every time we do something we first have to invent the methods and then we can do the experiment because nobody's done this stuff before. Thank you very much, Harry.
Thank you, Peter.
I'll just make a comment. The blue dots are methanogens. I just added this slide at the last minute because someone said that I didn't have my model methanogen in my pocket, which I often do to show people. So that's what they look like in rumen contents.
Yeah, right. I just have one very quick question before I move on to questions from the panel. And the app failed on my phone, but I've now been given another phone that seems to have got some of the questions from the floor. But just any very quick question that's specifically to Peter on his presentation.
Any thoughts about using mRNA technology to inject into the salivary glands to produce the antigens?
We have been looking at that. That was raised the last conference, and our discussions with the methane vaccine think tank pointed out that one of the issues is the pattern of glycosylation of the proteins. You'll get a mammalian glycosylation pattern which won't look anything like a methanogen one, so particularly for protein antigens, that's what you have to produce with an mRNA. That could be an issue. We've since talked further about that, and it's something we're definitely looking into. So it's not off the table.
Thank you, Peter. If you'd like to sit down and then we can ask the questions.